Okay, so today we are going to discuss part 1 of chapter 19, which is the chapter on oxidative phosphorylation. And in this chapter, what you are really going to learn is how when the metabolic fuel was oxidized, and in that process you had reduced electron carriers, how do those electron carriers transport their electrons to respiratory chains, and in that process how ATP is synthesized. Okay, so this is almost taking reduced form of NAD plus, FAD, FMN, and bringing those electrons to the respiratory chains and producing ATP. Now, I will briefly discuss the importance of reduction potential. I know that you are aware of reduction potentials and the equations that govern how reduction potentials are calculated. But I want to briefly go over that so that you are aware how typically redox reactions are managed. So there is a relationship between reduction potential and free energy, and we will briefly discuss that. And then, of course, we will dive into talking about the electron transport chain, right? And when we are actually transferring electrons from those reduced carriers to those electron transport chain, we actually create a proton gradient in the intermembrane space of mitochondria, and that intermembrane gradient of protons drives the synthesis of ATP. And so where do electron transport chain reside, and how oxidative phosphorylation happens, right? So as I said, electrons that are carried by the reduced coenzymes such as NADH and FADH2, they are actually passed through a chain of proteins that are embedded in the inner mitochondrial membranes. Okay, and so when the electrons are transferred to them, as I said, you generate a proton gradient, and that proton gradient runs downhill. Okay, so proton gradient that you create is a form of energy. Okay, so it runs downhill through F0 F1 synthase to synthesize ATP. F0 F1 ATP synthase. You have seen the structure of that before. Okay, so it all happens in or around inner mitochondrial membrane. With that, I want to show you how mitochondria is so fascinating. It's really one of the most important organelle in our cells, right? It's shown here on the left-hand side. You can see like a shoe. It looks like it's cut open from the top. And you can see that there is an outer membrane. Outer membrane of mitochondria is freely permeable to small molecules and ions. It is the inner membrane that is actually fascinating. Okay, so inner membrane, as shown here, okay, is formed from this very specialized structure called cristae. Okay, and I'll talk about that. I'm showing you on the right hand side. But the inner membrane is the very unique one, and it is impermeable to most small molecules, including protons and ions. Okay, so protons cannot just diffuse in and out, although they're really small molecule, right? And it's the inner membrane that contains the respiratory chain, which would be the complexes 1, 2, 3, and 4. A very important transporter protein called ATP-ADP translocase. ATP-ADP translocase is a transporter protein. It's actually an antiporter that allows ATP that has been synthesized in the mitochondrial matrix to go out of mitochondria, and it allows ADP to actually come into the inner mitochondrial membrane, right? So it allows the transport of ATP and ADP. And so, so ADP is brought from the cytoplasm to the mitochondrial matrix, while ATP is transported out from mitochondrial matrix to cytoplasm. And if you really look hard, you will notice that almost 10%, and that's quite a large number, 10% of the proteins that you have embedded in the inner mitochondrial membrane are ATP-ADP translocases. Okay, so it's a secondary active transporter, right? And as I said, it's an antiporter, which basically one solute moves in one direction, the other one goes in the opposite direction, right? And it is an active transport, secondary active transport. And so you know what secondary active transport is, right? One species of the solute will move along its electrochemical gradient, and that allows the other species to move against its electrochemical gradient. That's why it's active transport, although it is secondary active transport. Okay. And so, and you also see that you have, ATP synthase. We just talked about that, right? We have shown the structure of F0, F1 ATP synthase. And then you have other membrane transporters. What about the matrix? Matrix is shown here, in this yellow portion. They're actually, if you pay attention, they're showing you this yellow. It's almost like an island, right? Cristae are this very unique structure, okay, that literally, are this membrane, inner membrane that actually folds into these structures. And so you can say that it provides a much higher surface area in that little space. If you look here in the right-hand side, the cristae are shown here in different colors. This is actually a real computer-generated image of mitochondrial matrix. Mitochondrial matrix is all that dark space that you are seeing, all right? So anyway, so the matrix itself, contains pyruvate dehydrogenase complex, right? Citric acid cycle enzyme, fatty acid beta oxidation enzymes, right? Amino acid oxidation enzymes, of course, many other enzymes, all these ions, and tons of soluble metabolites. So, and of course, they are also showing you porine channels, which will allow the water movement from inside to out, and you have ribosomes, etc. Right-hand side, as I said, is computer tomography, computer-assisted, tomography image, and this is actually produced from X-rays. They basically utilize X-ray, and you get an image, you process it using computers. And if you do that, you can actually, get a slice, like virtual slices of any, three-dimensional species that you can actually look at. Okay, and it allows you to see. Maybe you don't have to cut the organelle to be able to see that. It actually literally slices. X-ray can penetrate and scan for the surfaces and show you the image as if it was sliced. Okay, very cool, very cool. And so, so mitochondria again, number of mitochondria will depend on what kind of cells we are talking about and what those cells' energetic needs are, right? So if you have, for example, active skeletal muscles, you may have thousands of mitochondria in those cells. But for example, red blood cells, which which are not active, their primary purpose is to transport molecular oxygen to respiring tissues. They do not contain any mitochondria, right? So just so you know, this is so important organelle in our cells and in our body. So with that brief introduction, I want to actually jump to talking about reduction potential. Okay, so one thing you will notice is that all electrochemists have adopted this convention, as I have referred to this in my earlier lectures, writing all redox reactions in terms of two reduction half reactions. Okay, so each redox reaction, for example, you will have a reduced donor, right, that will transfer its electron to oxidized acceptor. And so that's your overall redox reaction, right? And so electrons, so when you write your redox reaction as two half reactions as a reduction reaction, then you have one thing to worry about. You always look at standard reduction potential. Okay, so when we are talking about standard electron reduction potential, electrons are typically donated by the half reaction with the more negative reduction potential, okay, and are accepted by the reaction with the more positive reduction potential. So remember, we are only talking about reduction potentials, and that actually really has straightened out confusions that actually used to exist in the literature and among scientists and students. Okay, and standard free energy is linked to the standard standard reduction potential via this equation. Delta G naught is equal to minus NF delta E. And the way you calculate delta E is E0 of acceptor minus E0 of donor. And that's what I'm showing you right here. Okay. So you have an equation that looks like this is related to the free energy. And you have seen this equation in your general chem classes. Okay. F is a Faraday's constant, right? Delta E naught is the difference in the reduction potential between the donor and acceptor species, right? And N is the number of electron transferred. So this is something that you know. So if your delta E value, E naught value is a positive number that you can calculate by following this equation, then your delta G standard will be negative, and that redox process will happen, will be thermodynamically favorable, right? So because that delta G will be a negative number, right? So that's something that you should know. I know that you know this already, but it's very, very important to know. So how is it useful in a real context of today's lecture? This table actually lists you, so you'll see that they have listed bunch of redox reactions, but these are all half reactions, okay? Half reactions as seen here, right? So each of these half reactions, on the right hand side, they're listing you standard reduction potentials, okay? Cool. So you can see molecular oxygen being reduced to 1 mole of water. Standard reduction potential for that half reaction is 0.816 volt. Cool. Cytochrome C, various cytochromes, and you will see some of those today. Cytochrome C, for example, which we will talk about today. You can see that reduction of cytochrome C is 0.254 volt. So it is less positive, right, than molecular oxygen reduction half reaction. Very, very important conceptually for you right now, what I'm suggesting. Similarly, I'm just trying to see pyruvate to lactate, fumarate to succinate half reaction. as written as a reduction. You see, all reactions are written as a reduction reaction, right? Reduction half reaction. So that's why this is all standard reduction potential you are seeing. So remember what I suggested in the previous slide. Electrons are always donated by the half reaction with more negative reduction potential and are accepted by reaction with the more positive reduction potential. So if you look here all the way, NAD plus, FMN reduction, all those reduction potentials you see are much more negative than the molecular oxygen standard reduction potential, right? So in principle, you can take electrons from any of the reduction half reactions shown here at the bottom and should be able to transfer those electrons under standard conditions to molecular oxygen. Why? Because molecular oxygen standard reduction potential is more positive and the ones that are down here are more negative. And you are noticing that electrons are donated by the half reaction with the more negative reduction potential and are accepted. So that makes sense if everything is in the standard conditions, right? But remember, many of these coenzymes, for example, NAD plus, FMN, they are also part of a protein structure. So sometimes their reduction potentials are modulated by the environment of the protein, okay? But the list that you are seeing here is the standard reduction potential when they are not surrounded with proteins and other structures. But still, looking at this overall, you can see that it does make sense now. How you can actually take electrons from a reduced fuel like glucose, fatty acid, for example, transfer those electrons to reducing equivalents like FAD, FMN, or NAD plus, right? And those guys in their reduced form should be able to transfer those electrons to molecular oxygen without violating any rule of thermodynamics, right? Because that's thermodynamically favorable. I just told you delta G is equal to minus NF delta E. So that's something that you should know. Very, very important conceptually. Anyway. So with that brief introduction, now let's look at electron transport chains, how they are organized. It turns out that there are 4 protein complexes and they all reside in inner mitochondrial membrane. There is a lipid-soluble coenzyme called quinone or ubiquinone, and they are often written as like this here, UQ, Q, or CoQ, or sometimes CoQ10. They all refer to the same lipid-soluble coenzyme. And sometimes people use them without being formally telling you whether they are in where this lipid-soluble coenzyme is in the reduced state or oxidized states. But that's what they're referring to, okay? Remember, Q, UQ, CoQ, CoQ10. And I'm sure you have heard about CoQ10 on TV on some ads. So it's a fat-soluble coenzyme, which means it hangs out in the membrane. It is not happy otherwise. It will never go outside the membrane. Okay, this is a membrane-soluble coenzyme, lipid-soluble, fat-soluble. And then you also have a water-soluble protein called cytochrome C. Okay, cytochrome C has a prosthetic group. I'll talk about that. That actually is also involved in shuttling between different protein complexes. Okay, so coenzyme Q and cytochrome C, they actually shuttle electrons from these reducing carriers to between complexes, complex 1, 2, 3, 4. And in general, no laws of thermodynamics are violated in this process. Okay, remember, they are still upheld. So electrons generally fall in energy through the chain. For example, from complex 1 and 2 to complex 4. Okay, so they are downhill. What are those 4 complexes? That's what is shown here in this table. Okay, protein components of this mitochondrial electron transport chain is shown here. 4 complexes. First one is NADH dehydrogenase. It's the most massive one, 850 kilodalton molecular mass, and it has 43 peptidyl units. Okay, the ones that you're seeing here in parentheses are the ones that exist in bacterial enzyme. You don't have to worry about that so much. And they're showing you what kind of prosthetic groups they need for their function. Okay, so FMN and iron sulfur center is what you need for NADH dehydrogenase. Second complex is succinate dehydrogenase, and it has 4 peptidyl units, 140 kilodalton in mass, and it uses FAD and iron sulfur center as prosthetic groups. The third one is ubiquinone cytochrome C oxidoreductase. Its molecular mass is about 250 kilodalton, and there are 11 peptidyl units, and it needs hemes and iron sulfur center as prosthetic groups. Cytochrome C is not part of the complex 3, but it is actually involved. Okay, it moves between complex 3 and 4. Okay, as a freely soluble protein. Okay, and you will see that. And cytochrome C is, a small protein of only 13 kilodalton, and it's only one polypeptide unit that actually has heme as the prosthetic group. And the fourth complex is cytochrome oxidase. Cytochrome oxidase will oxidize the cytochrome C in particular. Okay, and that's 160 kilodalton, and it has 13 subunits. Contains hemes and 2 types of copper centers, copper A and copper B. And we'll talk about these individual electron transport chains going forward. This is all today's lecture is devoted to. Right? All right, so it turns out that I already am just telling you that you have certain types of groups, prosthetic groups that you will need to actually take electrons from those reduced electron carriers and eventually help transport them to molecular oxygen, right? And so the mitochondrial respiratory chain has, a series of sequentially acting electron transport carriers that contain specialized prosthetic groups. And those prosthetic groups are capable of doing either 1 electron transfer or 2 electron transfer reactions, redox processes. Okay. And what are those prosthetic groups? FMN, FAD, quinone, as I just showed you, 2 types of iron containing proteins, for example, cytochromes and iron sulfur proteins, right? So let's look at the quinone, because that's something that you have not seen so far, okay? Or also known as ubiquinone, okay? As I said, these names are sometimes loosely referred. It's also known as coenzyme Q10, okay? It also has other names, like people will say coenzyme Q or CoQ10 or only Q10. But basically, it's a coenzyme that exists, in all types of cells, okay? And basically, if you look at the structure that I'm showing you on the left-hand side, it is a 1,4-benzoquinone, okay? So quinone, you can see Q refers to that quinone moiety that you're seeing here, right? Chemical group. And what is the 10? CoQ10. What is the CoQ10? You can see that it has a nice hydrophobic tail of isoprene units on one side. And you can see that there are 10 isoprene units. And so that's why the name 10, CoQ10. Okay. And so on the top, I'm showing you this structure of ubiquinone that is in the fully oxidized form, right? And so in the fully oxidized form, it exists as a quinone. Now, ubiquinone can do both 1 electron and 2 electron redox processes. And so if it is doing 1 electron reduction, you'll end up generating a semiquinone radical that is shown here as QH dot. And if you do one more, then you will convert ubiquinone Q to QH2, which is ubiquinol, right? Those two ketone group became hydroxyl here, right? Ubiquinol, fully reduced, also known as QH2. So now from now onwards in this lecture, I'm just going to show you Q or QH2. So you know what we are referring to, right? What about cytochromes? We didn't talk about cytochromes too much. So let's just briefly look at this, okay? And there are 3 different types of cytochromes, okay? There is this B-type cytochromes, right? But anyway, first let's look at these cytochromes overall. Do you see any similarities? Yeah, you see similarities that you have this 4, this is like almost porphyrin type of rings, conjugated pi rings, right? That is made up of 4 pyrrole rings, right? And so that's what you're seeing here in the pink, right? So you have 4 5-membered nitrogen containing ring, right, in the cyclic structure here. And that cyclic structure you have seen is known as porphyrin, right? And in this porphyrin ring, you see that central, and in the center, there is an iron atom, right? And that iron atom is coordinated with those pyrrole rings, right? It can exist either as in +2 state or +3 state. So if you talk about B-type cytochromes, which are these proteins that will contain this type of prosthetic groups, protoporphyrin 9, you have seen this. You know, this protoporphyrin 9 is found in B-type cytochromes, and you have seen them in your structure of hemoglobin and myoglobin. That's the one here in the left. Heme C, which is found in C-type cytochrome, right? You can see that Heme C is where it's covalently bound to the protein of cytochrome C through these 2, thioether bonds. And those thioether bonds come from the 2 cysteine residues of the protein itself. So that would be C-type cytochromes. That's how heme is found in those. And then you have heme A, which is found in A-type cytochrome proteins, right? Has a long isoprenoid tail here attached to one of the 4 pyrrole rings, right? And you also see an aldehyde kind of functionality on one. So they have different functions, but overall this is how the prosthetic groups look like in these 3 types of cytochromes. Now, one thing you have to remember, and that's why I have it in red here, is that cytochromes are 1 electron transfer agents. Okay, cytochromes and iron sulfur proteins are both 1 electron transfer agents. They can only do 1 electron transfer processes, redox processes at a time. Okay, and so going back to so yeah, if you want to actually do lots of oxidative phosphorylations in certain types of cell, you can see that your muscle cells, for example, skeletal muscles, will need good amount of lipid soluble ubiquinone, so you can actually accept electrons and burn that energy, right? Anyway, so let's keep moving forward and introducing you to iron sulfur center, because you have not seen iron sulfur centers either in your biochemistry class or organic class. And so these iron sulfur centers are these iron sulfur proteins, okay, and they come in different flavors. And those 3 flavors are at least shown here in A, B, and C, where you see a single ion is coordinated with 4 cysteine residues. These cysteine residues are organic cysteines, okay. So this is a standard 1 ion and 0 sulfur system, if you want to call it, but that's, you know. In B, what you are seeing is that you have 2 iron atoms coordinated with 2 inorganic sulfur right here in the center, and then each coordinated to 2 cysteine residues, organic sulfur, okay. So this one in the B is called 2 ion 2 sulfur center, okay, because you have simply 2 ion, you can see in the pink, and 2 inorganic sulfur in the middle. That's why 2 ion, that's how nomenclature works, okay. And so, and in C, you are seeing 4 ion 4 sulfur centers. You can see 4 ions, right, in the cube, and they are coordinated with 4 inorganic sulfur units. So you can see there 1, 2, 3, and 4. So those are 4 ion 4 sulfur centers. So in this nomenclature, what I'm trying to refer to you is that organic sulfurs don't count in this nomenclature, okay. So what is the difference between iron sulfur proteins and cytochromes? In iron sulfur proteins, you can see that iron is present not as a heme, but in association with inorganic sulfur atoms and organic sulfur atoms, like from cysteines. And I'm just showing you, just to show you a structure of this protein called ferridoxin, which is a 2 ion 2 sulfur protein. This is actual crystal structure, and if you want, you can just type that code 1FRD in PDB data bank, and you can look at the structure, you can rotate it, and you can actually see that indeed it has 2 ion, it is a 2 ion 2 sulfur protein because you have 2 inorganic sulfurs bound to it. So obviously they can do 1 electron transfer reactions. They can participate in 1 electron transfer reactions like cytochromes. And so, but you know, their standard reduction potentials could vary depending on what is the environment of that iron sulfur center. And as a student of biochemistry, you can appreciate that, I am sure, because the reduction potential can change depending on the environment of the redox center, right? Okay, so that's the introduction to iron sulfur center, quinone, and cytochromes, that all of which you have not seen before. With that introduction, you are ready to dig a little more into the reactions of the respiratory chain. So they're showing you bunch of reduction reduction half reactions here, right? So you can see all half reactions are written in the form of reduction. This is how the convention now is. And you can see at the bottom, they're showing you the molecular oxygen half reaction, half a molecule of oxygen plus 2 molecule, if you want to say not molecule, but 2 equivalent of half equivalent of molecular oxygen, 2 equivalent of protons, and 2 electrons will give you 1 mole of water. You can see standard reduction potential for that reaction is 0.81 volt, right? So if you actually look at all the reactions, all the half reactions that happens in the respiratory chains that are listed here, you can see that they progressively fall from negative on the top to more positive at the bottom. And therefore, you know that electrons can be transferred from the top to the bottom half reaction without any problem, right? No laws of thermodynamics are violated here. Now, do I want you to memorize these reactions, every reactions? No. Do you want me to, am I expecting you to memorize the half standard, half reduction potentials? No, of course not. But what I want you to know and appreciate is that, yes, indeed, this looks like the half reactions of the respiratory chains, and those half reactions look like they are nicely set up in the proper energetic order so that electrons can indeed be transferred to molecular oxygen without violating the laws of thermodynamics. Okay, so that, let's look at the individual transport chain. Okay, so first we will talk about the NADH dehydrogenase. NADH dehydrogenase is also known as NADH ubiquinone oxidoreductase. If you see the name NADH ubiquinone oxidoreductase, you know that likely this enzyme will be taking electrons from NADH and transferring that to fat-soluble coenzyme ubiquinone. Yeah, that's exactly what it does. But there are various names. And it basically allows the electrons to be transferred from NADH to ubiquinone. So you can see on the left hand side is NADH. Electrons will be transferred from NADH to FMN to iron sulfur center and eventually to quinone, ubiquinone. And so that's why you are seeing ubiquinone in reduced form, which is ubiquinol, QH2. That's what they are showing you. That's what these arrows mean. That's just showing you electron path. And you know that NADH has one pair of electron, right? Two. So for whenever this complex transports two electrons from NADH to ubiquinone, it transports 4 protons outside of the inner outside into the space, which will be the intermembrane space of the mitochondrial membrane. So that's what this line means. 4 protons transported out from mitochondrial matrix into intermitochondrial space, like between the outer mitochondrial membrane and inner mitochondrial membrane. Now you can already think, if you are thinking about this, then outer mitochondrial membrane will not be leaky to protons, although it allows some ions to come in, but not protons, because it's like it stores energy there in that space in the form of this proton gradient, right? Pretty cool. Now, let me talk to you about this complex one, okay, in little bit more details, okay? And I know this is a very, very busy slide for you, but I will go through it so you actually can learn something new and enjoy, okay? So hopefully will not be going too fast. The structure of complex 1 is shown here on the right hand side, and you can clearly see that there is an N-2 arm of the NADH dehydrogenase, and then there is another arm, what we call membrane arm, that is actually right there in the membrane. Okay, it turns out that the matrix form of the NADH dehydrogenase crystal structure has been solved at the atomic level. So you can look at all these, centers, redox centers, how they are organized in this protein. Beautiful, beautiful structure. Okay. So you can see I'm showing you that NADH will come, right, and transfer, and before I go, you are seeing the membrane right here in yellow, right? On top of that membrane is where there will be intermembrane space. it's known as P side also. People will just write P side. You are supposed to know that P side means they're talking about intermembrane space, which is the space between inner mitochondrial membrane and outer mitochondrial membrane. P side, why P side? Because that's where protons go. And the matrix side is always referred to as N side. So just note that. So what will happen? NADH will come and basically transfer the 2 electrons to FMN center, shown here. And those electrons then flow through a series of iron sulfur centers, eventually going into the membrane and transferring themselves to the lipophilic electron acceptor ubiquinone Q. And so ubiquinone becomes reduced form of ubiquinone, which is ubiquinol, QH2. And in that process, conversion of quinone Q to QH2 sucks up 2 protons from the matrix sides. Okay, not only it sucks up 2 protons from the matrix sides to make QH2, but also it transports 4 protons from the N side to the P side. Okay, concomitant transfer of 4 protons from N side to the P side. So you can see what's going on, and that's what they are showing you. When you look at the crystal structure, you can see that these series of iron sulfur centers are beautifully organized, by which electrons just hop in a downhill energy fashion and eventually reach to Q, which is hanging out in the membrane space, and Q getting converted to QH2. Okay. And so the mechanism that couples the transfer of electrons and this proton transfer, pumping up the protons, mechanism is exactly not dissected yet, but it involves something like a Q cycle that we will talk in complex 3. Okay, so just hang in there. I'll talk about Q cycle when we discuss complex 3. And Q cycle is where QH2 participates twice per electron pair. Okay, remember, one QH2 molecule has 1 electron pair, 2 electrons. Okay, and so we'll get to that when we get to that. Okay, so that's what complex 1 does. It basically catalyzes 2 simultaneous but obligatory coupled processes. Okay, what are those 2 processes? Well, the first process is this exergonic transfer of hydride ion from NADH to eventually Q, right? That's an exergonic reaction. And that exergonic reaction is coupled to the second reaction, which is endergonic, which is the transfer of these 4 protons from the matrix sides to the intermembrane space. Those 2 processes are coupled. Okay, that's what complex 1 does beautifully. So if you look at the overall reaction, you can see here balanced equation is shown here, NADH plus 5 protons from N side plus ubiquinone going to NAD plus reduced form of ubiquinone, which is QH2, ubiquinol, plus 4 proton. Okay, they are trying to balance the charge in the P side. Okay, this proton that actually exists is the conserved energy. Okay, that's what is happening. Your energy is conserved in this proton gradient that this complex 1 is creating. Where is that energy coming from? NADH. You're stripping away of electrons from NADH to Q. And it turns out that I have a homework here. You are supposed to go and read the effect of some molecules like amytal. Okay, amytal is a barbiturate, and barbiturates are these medical agents that cause hypnosis. They also have sedative actions, and some people are suffering from severe anxiety, etc. They are utilized as drug. Okay, and they're not easy molecules to deal with. They can be very dangerous if you don't give the correct dose. Okay, so but anyway, and rotenone is a plant product that is used as an insecticide, right? And pyricidine A is an antibiotics. And it turns out that all three of these three molecules are inhibitor of complex 1. Okay. And the way they inhibit the function of complex 1 is that they block the oxidation of iron sulfur clusters of complex 1. They go and bind and basically shut down the flow of electrons through this complex 1. Okay, so just the complex 1 is affected by these specific molecules, not complex 2 or any others. Okay, so just something to know in the side. Okay. Oh yeah, so let's talk a little bit about how scientists were able to actually get a head start in treating Parkinson disease, or actually finding a cure for Parkinson disease. Okay, so it turns out that in 80s, people were not only listening to psychedelic music, but they were also heavily involved in using drugs. And so when the drug demands were high, black markets were producing a copious amount of synthetic heroin, and it was causing trouble on the street, because people will, it's black market, so people didn't know what they were smoking or whatever you do with heroin. So yeah, so a lot of people were getting this paralysis, and they can't function, their legs are all gone, arms are totally dysfunctional, and so it was a real tragedy. And that tragedy was happening because there is a molecule that was actually found in this illegal drugs called MPTP. Okay, MPTP stands for 1-methyl-4-phenyl tetrahydropyridine. I'll show you the structure, okay, in a minute. I think it's in the next slide. But basically, it's a compound that is found as a contaminant in this synthetic heroin. And when this compound was going into the brain, it basically was rapidly getting converted to another molecule called MPP plus. Okay, MPP plus is again 1-methyl-4-phenyl pyridine. And this MPP plus was inhibiting complex 1. It's a very potent inhibitor of complex 1. Okay. And so people were noticing that when it was inhibiting complex 1 in the brain, that area of inhibition was happening in the same location where Parkinson's patients are being affected. Okay, so that's the same region of brain that gets deteriorated in Parkinson's disease. And so when they were actually figuring this out, they were able to see that if you actually take these patients and treat their tissues with L-dopamine, etc., they could actually restore movements. Okay, so there were a lot of research related to that. In fact, you can actually implant fetal brain tissues, and that could also work, in those regions if you actually implanted a fetal brain tissues. So, something good came out of this. Okay, and this really led to revolutionize the use of tissue implantations to treat certain neurodegenerative diseases. So from bad things, something nice came out. But basically, that's the structure of MPTP. And this brain has this enzyme called monoamine oxidase B, that will take MPTP and convert it into this very stable MPP plus. You can see that you have now 2 aromatic rings as opposed to 1 here. And so this molecule MPP plus will actually go and get enriched and basically cause cell death in this region of the brain. I'm sure many of you are neuroscientists, substantia nigra, and that would be the cause of this paralysis. So yeah, that's a little bit of medical mystery and how it was solved. Now with that, let's talk about complex 2, okay, which is the succinate dehydrogenase. So complex 2 is, as you know, and I am pretty sure now, something must be ringing in your mind, right? Oh, I have seen succinate dehydrogenase. Where did you see this enzyme? In citric acid cycle. Remember, succinate to fumarate to malate to oxaloacetate reactions that happen many times in several metabolic pathways. Yeah, you utilize succinate dehydrogenase. It's the same TCA cycle enzyme. And it's the only membrane-bound enzyme of the citric acid cycle. I didn't tell you in the citric acid cycle, but now you know. Succinate dehydrogenase is the only membrane-bound enzyme of the citric acid cycle. What does it do? It basically takes succinates and reduces it, eventually to fumarate via phenol. Okay. So FAD is the prosthetic group that actually is found covalently bound in succinate dehydrogenase, right? And it has 4 different subunits, A, B, C, and D, and 5 different types of prosthetic groups. Okay, so you have 3 different types of iron sulfur clusters. So you have 4 iron 4 sulfur centers, you have 3 iron 4 sulfur centers, you have 2 iron 2 sulfur centers, right? So that's 3 type of iron sulfur clusters. You have FAD, that 4, and the 5th one is not shown here and not utilized here, but exists in succinate dehydrogenase. It has stayed there through evolution, although it does not directly participate in the processes of complex 2. And you'll see this fascinating, beauty of proteins and how cool chemistry they do. Okay. So net reaction of succinate dehydrogenase is you take succinate and transfer those electrons to fat soluble coenzyme Q, generating fumarate and reduced form of QH2. Okay, so let's look at this, how this complex looks like. Okay. Complex 2 structure has been solved. So we know at the atomic level how things work here. Beautiful, beautiful. So if you look here, right, you can see clearly that there are 4 subunits, A, B, that sits on the matrix side, N side, and then you have unit C and D that are membrane bound, right? Cool. So those are the A, B, and C and D. Now in A, right, just behind, you can see they are showing you FAD moiety right here in the pink or whatever color you are seeing. Behind that FAD is where the substrate binding site is. Okay. And that's in subunit A right here in gray. So where does succinate binds? Right behind FAD in subunit A. So blue arrows are the arrows where electrons are moving. So pay attention to those. Okay, very, very important. And I want you to always be focused on your blue arrows here. They're so carefully drawn, they mean a lot actually. All right. So that's subunit A, succinate, and then you have FAD on top. And you can see FAD. You can't see succinate, but that's behind FAD. Then you are seeing these yellow balls that are iron sulfur centers, right? Right? So subunit B has these 3 iron sulfur centers shown here in the yellow. Okay? And one of the yellow here, if you notice, I don't know why they couldn't choose a different color, is also in subunit B, but it's kinds of at the interface of the membrane and matrix, which is ubiquinone. So this right here is ubiquinone. You see? Ubiquinone is bound to the subunit B at the interface. Cool? So you can see here 3 iron sulfur center and ubiquinone in subunit B. Right? And then you have this heme B, which is shown here in the membrane in red or pink, that is sandwiched between C and D domain of the protein. Right? You can see that nicely sandwiched there. And you also see 2 molecules of phosphatidyl ethanolamine that are also found in the crystal structures, and scientists are still trying to figure out what role they might be playing in the function of complex 2. But you will see the beauty of complex 2 in a minute. I will mention how precise this unit works together. Beautiful, beautiful. Okay. But one thing I want you to notice is that when you see the flow of electrons as shown here by blue, you see that from ubiquinone, once the electrons are transferred to ubiquinone, the ubiquinone in the reduced form diffuses in the membrane, QH2. Heme B doesn't even come in that electron flow path. So what's it doing there? What function Heme B plays? That's interesting, right? It turns out that Heme B is not on the main path of the electron transfer, as you can see here in the crystal structure beautifully. It comes after actually electrons are passed to ubiquinone. So what is it doing? It turns out that this is a protection mechanism in the electron transfer pathways, because if electrons are not properly transferred and if some of the electrons went astray, unchecked, then those electrons can create radical species, and then radical species can form another radical species, and that could lead to mutations in the protein and DNA, and that could give rise to tumors. Okay. That electrons can also give rise to other types of what we call reactive oxygen species, ROS. Okay. And the ROS are really, if they're not in check, they can give rise to tumors. So people's hypothesis is that Heme B is placed there as a protection mechanism so that you do not form ROS species by electrons that go astray. Okay, that's the function. And how do you validate that? How do you validate that hypothesis? It turns out there are some people who are born with a condition, right, called paraganglioma. I think it's passed from the parents, so it's a hereditary condition. Okay. And this paraganglioma is what? It's actually a benign tumors of head and neck. Okay. And it is, it is generated in this, carotid body of head and neck. It's a benign tumor. It's not cancerous. Okay. Now, what is carotid bodies? Again, we're not going to go into biology, but basically this carotid body, is a group of these chemoreceptors that sense how much oxygen is there in your arterial blood that is flowing through your head and neck. Okay. Yeah, it's amazing. Yeah, so this you always are, testing the partial pressure of oxygen, so you can regulate the oxygen supplies to blood, and things like that. But carotid body, is these supporting cells that are there to sense the amount of molecular oxygen in the arterial blood that are flowing through head and neck. But anyway, it turns out that if you have a mutation in complex 2 subunits, right, near especially where the heme B binds or where this quinone binds, right around there, okay, then you can suffer from this benign form of tumor. I have seen people actually as a kid, and not one or two, but few at least, that these people will have like a big, massive, amount of tissues just growing in the back of their neck or back of their, head. And they lived normal life. It was not cancerous. Okay, so so the people that those tissues are regenerated, I think, because if that you have mutations in that region that will allow those electrons that went astray, didn't get transferred faithfully to ubiquinone. Those cells will actually will divide, proliferate, and give rise to these benign tumors. Cool, right? Think about what beauty of protein complex playing such an important role in our function with the help of several prosthetic groups and this beautiful organization of molecules, right? All right. I know we went a little bit overboard, but let's keep moving. With that, let's go and talk about the complex number 3. Oh yeah, just take a deep breath because this is going to give you a little headache. Okay, but be with me. I will try to repeat this, I think, in the next class, especially the Q cycle. It's fascinating how things happen. So complex 3 is ubiquinone cytochrome C oxidoreductase, right? And so in this complex 3, what you do is that you collected electrons from complex 1, right, and complex 2 in the form of reduced ubiquinone, which is QH2, which is ubiquinol, right? And so you must transfer these electrons, keep going, and you bring them to complex 3 and pass them on to this 1 electron soluble protein carrier called cytochrome C. Okay, so ubiquinol will pass those electrons to cytochrome C, and in that process it will also pump protons in the intermembrane space. And based on a lot of scientific experiments, people have understood how this complex 3 might be working at the molecular level. Okay, crystal structure of complex 3 has been solved, I think, in mid-90s. Okay, so we know at the molecular level what goes on in this. But Q cycle is a working model that explains the stoichiometry of proton pumping into the intermembrane space per equivalent of electrons transferred from ubiquinone to cytochrome C. Okay, so there are experiments involved, and that's how people have come up with this Q cycle working model, how complex 3 might be working. Okay, so principal component is B cytochrome. Okay, and B cytochrome has this heme BL and BH. So let's look at the reaction itself. You take the reduced form of Q. Reduced form of Q will have 2 electrons. So therefore, you will need 2 molecules of cytochrome C in the oxidized form to accept 2 electrons from QH2, right? And in that process, you have generated 2 reduced form of cytochrome C. You have pumped 4 protons in the P side, and you have consumed 2 protons from the N side in that reaction. That's the net reaction of complex 3. Let's look at it, the Q cycle, how that operates. It turns out that complex 3 is a dimer. It works as a dimer, okay? And so imagine what the figure I'm showing you here. Don't get too bogged down. I will walk you through every step of this complex 3. So this is the dimer you are seeing in the membrane. You can see N side on the bottom, P side on the top, right? And then you are seeing various protein complexes. Remember, this complex has this line, 11 individual peptide units, right? And it has the prosthetic groups as hemes and iron sulfur center, right? So it is a massive complex. Cool. So how do electrons from QH2 get transferred to cytochrome C, and how protons are pumped is explained by this Q cycle. Okay. So if you look here, right, let's go on the left hand side. Okay, so you start out from the top. That's where I will start out. Start out from the top left, you see QH2, right? In the green, you can see that that's the cytochrome B. Okay, cytochrome B having 2 heme center, BL and BH, shown here in the pink. Cool. In the front, cytochrome B protein with 2 hemes, BL and BH, shown here in the pink. Pay attention to that. Okay. So the path of electrons, let's start from one reduced QH2 molecule. Okay. So QH2 molecules will be coming to cytochrome B, where it will transfer 1 electron through this risque iron sulfur center, 2 iron 2 sulfur center, to cytochrome C that has a heme C, shown here in purple and pink. Okay, so they're showing you purple. You can see that that's your cytochrome C protein. It is in the intermembrane space. It is a water soluble protein. Okay, so 1 electron from QHT goes through iron sulfur center, right, to cytochrome C. The other electron flows down, as you are seeing, through heme BL and heme BH to an oxidized molecule of Q. Oh, interesting. I thought we were supposed to take the electrons from QH2 and transfer them to cytochrome C. Why would we transfer them to Q and try to reduce Q again to QH2? It doesn't make sense to you. It shouldn't. But these are experimental findings, and that's on which this working hypothesis is built. And the experimental findings are how many equivalents of proton are being pumped. People have done very careful experiments to come up with this cycle. So believe in this, but this is what is going on here. Okay, so you take Q on the N side, right? And you transfer 1 electrons to generate this radical anion. On the P side, you transfer 1 electrons to cytochrome C. Now cytochrome C, remember, is only one electron carrier. Once cytochrome C has accepted an electron, is in the reduced form of cytochrome C has very little affinity for complex 3. It will dissociate and go and bind to complex 4. It has higher affinity for complex 4 in the intermembrane space, reduced form of cytochrome C. Pretty cool. Think about how cool this is, right? Oxidized form of cytochrome C will bind to complex 3 on this, the way it's shown here in purple and pink, left hand side. But once it accepts electrons, it dissociates. Interesting. So what happened here? You took QS2 on the left hand side, you transfer one electron there on the top to cytochrome C, you brought one electron to the bottom on the N side, you transferred that electron, not actually in the matrix, but just still in the membrane, to quinone, because remember, quinone is not found in the matrix, so you have to be careful there, to generate this radical anion. Cool? And in that process, you pump 2 protons to the P side in the intermembrane space. That's what happens here. All right, so what is the product of this first cycle here on the left, as shown here left? Well, you made 1 mole of Q and 1 mole of radical anion, right? 1 electron transfer. This is the one that is shown here at the bottom. So Q will diffuse right into the membrane. In the second subunit, because remember it acts as a dimer, second subunit, again you will do the same thing, but not exactly. You'll take a molecule of QH2 and you transfer 1 electron on the top again to cytochrome C, right? And the second electron you bring and basically take that radical anion and reduced it fully to reduce form of ubiquinone, QH2. But in that process, you will need to suck 2 protons from the N side to make QH2. Pretty cool, right? So the end product on the right hand side of the cycle is that you brought in 1 molecule of QH2 and the product going out is 1 molecule of QH2 and 2 protons from the N side. And the product is that you generate 2 protons, you pump 2 protons on the P side, you generate 1 mole of Q and 1 mole of QH2. Okay, complicated, but does it really explain something? Okay, so here is what they have done for you, okay? What they have done is they have actually written the two redox half reactions, two redox reactions that's happening on the left and that's one that's happening on the right. Okay, so on the left, what are you seeing here? You can see reactant is QH2 and Q, right? So QH2 plus Q plus cytochrome C1 in the oxidized form is giving rise to Q dot negative charge, which is radical anion of Q, right? Plus in the product, you are producing a molecule of Q, again, you can see here. So plus Q, plus 2 proton on the P side, so 2 HP plus of course, you'll have cytochrome C that is being reduced. That is going to go now to complex 4 and transfer those electrons. So that's on the left hand side. On the right hand side, you are seeing the same thing, QH2 plus you are utilizing radical anion Q dot negative, plus you are utilizing 2 protons from the N side, plus a cytochrome C1 in the oxidized form, and generating a molecule of QH2, a molecule of Q, you pumping 2 protons on the P side, and then in that process you have reduced cytochrome C. So cytochrome C reduced. Cool. So if you actually did both of those reactions and wrote together and actually cancel things that can be canceled, your net equation is shown here at the bottom, where you take a molecule of QH2, 1 mole of electron, 2 electrons, 1 electron pair, 1 QH2, and you transfer those electron pairs to two molecules of cytochrome C1 that are in the oxidized form. And in that process, you are utilizing two protons from the N side, negative side, matrix side. The product is that you have one molecule of Q, two cytochrome C1 that are in the reduced form, and in that process you have pumped 4 protons on the P side. That's what the net reaction is, net equation. And you can see now it should make sense that this stoichiometry makes sense with this Q cycle that I just explained to you. You are able to utilize 2 protons from the N side. You are able to take a mole of QH2 and transfer those 2 electrons to 2 moles of cytochrome C. And in that process, you are able to pump 4 protons in the P space. So the Q cycle literally accommodates the switch between this 2 electron carrier Q and the 1 electron carriers cytochromes, right? B and C. And it also helps explains the measured stoichiometry of 4 protons that are translocated per electron pair that go through complex 3 to cytochrome C. So that's what it means. QS2 participates twice per electron pair. And what does that mean? Right here you can see QS2 on the top and QS2 again here on the right hand side. So it's participating twice per electron pair, transferring those 2 electrons to 2 molecules of cytochrome C. Pretty cool, right? It is fascinating. I hope you enjoyed this. You read this again and again, and you listen to the lectures, and it will make all sense to you. Okay. All right. That brings me to complex 4, which is cytochrome oxidase. And cytochrome oxidase has 13 subunits, and it has hemes and 2 copper centers as the prosthetic groups. But let's look at the, a little details of complex 4. Remember what I just told you. Now you took 2 electrons from QH2 and you were able to bring those 2 electrons to cytochrome C 1 electron at a time, because cytochrome Cs are 1 electron acceptor. These cytochrome Cs will go and transfer those electrons to ultimate electron acceptor in aerobic metabolism, which is molecular oxygen. So if you have to reduce molecular oxygen, you can see O2, you will need 4 electrons to generate 2 moles of water. Okay. So a complete reduction of a mole of oxygen molecule requires 4 electrons to generate 2 moles of water. So obviously, right away, it should click to you that that means 4 cytochrome C in the reduced forms will be needed to take molecular oxygen and convert that to 2 molecules of water, right? Okay, so let's look at complex 4. It turns out that complex 4 also transports protons across the intermitochondrial membrane, okay? And so there are total of 4 protons that participate in molecular oxygen reduction, and 4 protons are transported across in each catalytic cycle. And there are some inhibitors of complex 4 that will completely shut down oxidative phosphorylation process. Those are cyanide, azide, and carbon monoxide. Okay, and cyanide, azide, and carbon monoxide, they can, as I said, they can inhibit complex 4. And the way they do is that they actually inhibit the electron transfer by binding very tightly to those ions that are coordinated in cytochrome A, particularly cytochrome A3. Okay. So once they are bound, molecular oxygen cannot bind and it's all killed basically. But let's look at the flow of electrons here. All right. So 3 of the 13 subunits, remember I said there are 13 peptidyl units. It's 3 that are most critical for the electron flows. And those 3 units are shown here in yellow, blue, and purple. Yellow is subunit 1, purple is subunit 2, and blue is subunit 3. The green that you are seeing in the back are required, and they are the next remaining 10 peptides in the complex. They do play a role, but the electron flows are mostly communicated through complex subunit 1, 2, and 3 here. Okay. So if you look at the top, you will see that, yes, all right, you do need 4 cytochrome C to, make 2 molecules of water from 1 mole of oxygen. You can also see that 4 protons are being pumped into the intermembrane space as shown here. And 4 protons from the N sides are being utilized to convert a mole of molecular oxygen to 2 moles of water. All right. But it turns out that the cycle doesn't start with 4 cytochrome C bound to complex 4. Okay. It starts with 2 cytochrome C bound, one at a time. Okay. So 2 molecules of reduced cytochrome C will go and bind, right, and transfer their electrons to this binuclear copper A center. Okay, which is, you can see here they're showing you copper A right here in the green, right? That's binuclear center copper A in subunit 2. Cool. So they will go and donate their electrons, 1 electron at a time, to this binuclear center copper A right here. From here, the electrons will pass through this Heme A right here in the pink, as you can see. This center in subunit A is what we call iron copper center. You can see here iron copper center. There is a copper B, right, in green here in subunit 1. And then they are showing you Heme A. So electrons will make their way from copper A to Heme A of the iron copper center. Okay. And from Heme A, okay, so you can see here, if you pay careful attention, Heme A is on the right hand side and Heme A3 is on the left hand side, right, in yellow right here. So once electron pass through copper A to Heme A, and from Heme A, they are showing you directly that it goes to copper B. It doesn't go to Heme A3, by the way. Okay, it goes through copper B. Okay, copper B is part of that iron copper center. And so once that happens, okay, oxygen will go and now bind to Heme A3. Yeah, that's where oxygen binds. Oxygen binds to this Heme A3 of the iron copper center. Okay, of subunit 1. And then once molecular oxygen goes and binds, 2 electrons are transferred to it, and you form this peroxy derivatives, which is O2 with a 2 negative charge on it. Okay, 2 electrons. So molecular oxygen gets reduced to this peroxy derivatives. So it will be O2 with a 2 negative charges on it. It's not shown here, but that's what happens. Okay, and then the 2 more electrons will come from the top, from cytochrome C. Okay, so now you have all 4 electrons. those two next two electrons will convert O2 with 2 negative charge to 2 molecules of water with consumptions of the 4 proton from the N side. That's how you form 2 moles of water. Okay. When that happens, when you form that water molecule, is when 4 protons are pumped from the N side to the P side. Exact mechanism of this proton pumping is not fully elucidated. Okay, but that's what happens. When you suck the 4 protons to make water molecule from that peroxy derivative, is when 4 protons are pumped from the N side to the intermembrane space. Okay, and people have done lots of mutations, etc., to actually figure out exactly how these protons are pumped and how these protons are consumed, etc. But there are lots of details that are not yet known fully. Okay, so that's what the net reaction is. You take 4 reduced form of cytochrome C, 8 protons from N side, right? Plus molecular oxygen, you produce 4 cytochrome C oxidized, 4 protons are being pumped in the P side, and 2 molecules of water to balance the equation. That's how they have written. All right. So that's how molecular oxygen accepts electrons and gets reduced finally, right? So now it all makes sense to you, right? How this movement of electrons happens through the electron transport chains, right? And these 4 electron transport chains are quite independent, right? And but they are showing you here. And this is more or less a pretty good picture of how the electron transport chains are organized. These structures, individual structures that you are seeing here, are derived from different sources. Like some are human, some come from the pig enzyme, some come from the horse enzyme, and so on, or cows. But this is how overall the process looks like. Okay. You can see here that electrons, whether coming from NADH or from succinate, right? They reach Q through complex 1 and 2 independently. That's the key. Very important to note that. So you do produce complex 2 independently produce QH2, complex 1 also independently produce QH2. When complex 1 produces QH2, it utilizes 2 protons from the N side and pumps 4 protons in the intermembrane space. Complex 2 doesn't pump any protons in the intermembrane space. It simply transfers those electrons through Q to generate QH2. But once QH2s are generated, whether by independent means or together, they will come down to complex 3 as shown here. And sometimes students get a little bit knocked out with what they see here, but they are showing you 1 mole of QH2 transferring 2 electrons to complex 4. Remember, a full reduction of a molecule of oxygen is a 4 electron process, and when you do that, you generate 2 moles of water. Here they are showing you 1 mole of water. That's why they are using half an equivalent of molecular oxygen. Okay, so everything is half shown here, just so you know. But anyway, let me walk you through here. QH2 will come either from complex 1 or from complex 2 or both, will go to complex 3. Complex 3 will transfer those 2 electrons from QH2 to 2 molecules of cytochrome C. Remember, Overall cycle involves twice, remember, Q cycle, but they're just showing you one here. But they're showing you the stoichiometry of the reaction. You still consume in the Q cycle only 2 protons from the matrix side. So that's what they're showing you here. And you pump 4 proton in the P side. So that's what it means. Per mole of QH2, you consume 2 protons from the N side and you pump 4 protons from the N side to the P sides. 2 electrons do end up in 2 molecules of cytochrome C. And that cytochrome C will go and bind to complex 4, 2 at a time. So that's why they are showing you 2x. So it will either do that process twice. And then in this case, they are showing you 1 electron going at a time. So that's what they mean 2x. But basically, you know, as I said, it's half stoichiometry here. So you are seeing here. So you only need 2 electrons, right? Formally speaking. So that's why 2x, 1 electron at a time, 2 times you do that. Basically, will be able to generate 2 protons in the P side. You are consuming 2 protons from the N side. Remember, again, stoichiometry, don't get confused. If you actually were to put oxygen here and 2 molecules of water, you'll be using 4 protons here and you're pumping 4 protons there. So don't get thrown off by that. Cool, that's what happens. So in summary, we can see that complex 1 can independently transfer electrons from NADH all the way to molecular oxygen, which will be in complex 4. And when it does that, this is the stoichiometry of the reaction. Okay, you can see that you are pumping 10 protons in the P side. Okay, you are utilizing about 11 protons on the on the N side. If you are bringing complex 2, which means from succinate to complex 4, right? So succinate through FAD through to finally Q and to finally molecular oxygen, complex 4. This is your half reaction. You are starting out with FADH2, right? You are utilizing 4 protons from the, 6 protons from the N side, half a mole of oxygen. You are generating only 6 molecules, 6 protons in the P side, plus water. So look at this reaction, just look at it. NADH, which will come from complex 1, will go to complex 4, is producing 10 protons on the P side. Complex 2, which can independently bring reducing equivalents in the form of succinate through FAD, that can only produce 6 protons on the P side. So this difference in the number of protons that are transported into the intermembrane space reflects the difference in the ATP that is being synthesized. The higher the proton gradient, the more protons will flow through F0, F1 ATP synthase, and you will generate more ATP. This is why NADH on an average produces 2.5 molecules of ATP, and FADH2 produces only 1.5 molecules of ATP, because FADH2 starts from complex 2. And if you recall, complex 2 does not pump any proton into the intermembrane space, whereas complex 1 does. That's right there, your understanding of why you produce more ATP per NADH molecule versus per FADH2 molecule. And that's where we will stop. It's almost going to be full 2 hours, and we will start the next class from here. Let me stop the recording, and then we will